On to the next section of our Global One Health Conference entitled Genomics and Bioinformatics. Just going to provide a brief intro and introduce our plenary speaker. So the genomics and bioinformatics field is vast, and the implications on global health have been far-reaching. So since the early 2000s, with the discovery of the first mammalian genome sequences, the field has expanded at a logarithmic pace and opened the door to breakthrough discoveries, improving our understanding of cancer, zoonotic diseases, and many challenges that we all face across the globe. The field of genomics and bioinformatics has led to breakthrough approaches to better care for our environment, new technologies like CRISPR-Cas9, which have implications of their own that are far-reaching, and novel approaches to prevent diseases with new vaccine technologies and treat disease in a more logical and precise fashion with the emergence of precision medicine. So more importantly, this field has really taught us the relevance and importance of team science and international collaboration. And this has never become more clear than with this onset of our COVID-19 pandemic. In light of the broad nature of this field, we've invited our plenary and panelist speakers who will present their own work that illustrates the diverse and far-reaching implications of genomics and bioinformatics for global health. So to begin, I'd like to introduce our plenary speaker, Dr. Christina Tato. Dr. Tato earned her PhD in immunology from the University of Pennsylvania. As a postdoc fellow, her work focused on how transcription factors mediate host response or resistance to infection, regulate production of inflammatory cytokines, and affect the development of innate and adaptive immunity. At Stanford's ITI Institute, Dr. Tato focused on the tactical applications of systems immunology methods for studying human health and disease and for evaluating vaccine efficacy. She now leads the rapid response team at the Chan Zuckerberg Biohub and oversees planning and implementation of activities to strengthen global response efforts to emerging infectious diseases. The title of her talk is Putting Theory into Practice for Next Generation Sequencing Capacity. And Dr. Tato, I hand it over to you.
Great, thank you so much for that really nice introduction. Let's see, I'll just share my screen now. Oh, I have to wait till you stop sharing before I can.
Oh, sorry.
Oh, no, I guess it's good. There we go. All right. Great. Okay, thanks again, Robert, for the intro. And thank you to the organizers for inviting me to speak to you all here today. So I'm just going to start with a really brief introduction about the Biohub and then go into some of our work that we've been doing over the last 2 years and tell you a few stories about some of the groups that we've been working with. Oh. I cannot advance my... Not sure why I can't advance my slides. There we go. Okay, so the Biohub is a nonprofit medical research organization that is situated in San Francisco in California, and we work very closely with 3 research organizations that are also in the Bay Area: Berkeley, UCSF, and Stanford. We also work side by side with our sister organization, the Chan Zuckerberg Initiative. They are a separate organization, and they have a much broader reach in their work, not just in science, but also in education and in social justice issues. So we at the Biohub employ a basic science approach to understand the mechanisms of disease and then develop and apply new technologies to advance research and clinical practice. And we're led by our two co-presidents who themselves are very well known for their innovation in science and technology. And we have basically three areas of work that we're broken up into. So the first is our investigator program, through which we fund scientists across the three institutions that I mentioned to support their research. And we also have a number of different technology platforms. So they range from genomic sequencing to bioengineering and data science, as well as many others. We also have our own internal research programs. We have a cell biology program, which I won't speak to you about today. And we also have our infectious disease initiative, of which I'm a part of and my group is a part of. So in the infectious disease initiative, we really focus on improving detection and characterization of diseases and the pathogens that cause infection. We also work to identify new therapeutic targets that could be used for novel treatments and novel treatment development. And we also work towards improving strategies for prevention, such as advancing vaccine research. And really Respond, I put it in the center here because it really leverages the work across the BioHub and the different tools and technologies that we develop here in the Bay Area to take action in global health and to overcome challenges that exist there. So in large part, we help to build preparedness platforms by providing tools and standards and training to support sustainable capabilities in laboratories around the world. We are also here as a resource for rapid pathogen detection and identification for outbreak response efforts. And we also provide strategies for a One Health approach to public health surveillance. So really focusing on understanding the entire transmission dynamic of vector, reservoir, and population. So as part of our work, it's really in rapid response, we really try to understand the kinds of issues that exist in different lab settings. So especially in both not just limited resource settings, but in more rural settings where access is constrained. And so here on the left, I'm showing you some diagnostics that are very commonly used and often the first go-tos. But you know, there are limitations to these. They are tried and true methods, but they do require prior knowledge of the organism that you're looking for. So to pick the right stains or the right selection media. Also, their sensitivity and specificity can vary quite significantly depending on the assay. And so failure rate can tend to be very high, especially when you combine it with the fact that we are in a time, in a century-long time here of new emerging infections kind of increasing with every decade. Most notably, of course, are things like SARS and Ebola, as well as flu that have been causing constant and persistent issues for us these days. And so really this is where we hope that next generation sequencing can come into play. And so the idea here is to bring not just next generation sequencing, but also specifically metagenomic sequencing to bear alongside these more traditional microbiology techniques to really understand the full scope of the microbial landscape. And what this does is, and basically the reason why we think we can do this, and many people have started doing this already, is because the cost of sequencing, of course, is getting more affordable. Sequencing strategies and equipment is getting more portable, so you can do this in the field or in a mobile unit now much more easily than you used to be able to. But there are still a lot of the limitations. So training, of course, still needs to be done. Access to reagents and equipment is still a significant barrier for some, as well as doing the bioinformatics can be a hurdle for some groups. So we've been working very closely with engineers at the Chan Zuckerberg Initiative to develop analytical tools that are easily accessible and can be widely distributed for use. So one specific effort has been focused on metagenomic sequencing to understand again, the broader pathogen landscape in a biological sample. And here we've collaborated with CZI to develop a cloud-based infectious disease sequencing analysis pipeline called IDSeq that we can then make available to all the groups that we work with and beyond. So I hope through the talk you'll come to appreciate kind of the diversity of questions that metagenomics can be applied to, from straight up pathogen detection and identification, looking at etiologies of unknown infections, for example, or disease epidemiology to understand local outbreaks or patterns of antibiotic resistance, as well as many planetary health and aspects of research, so vector surveillance and doing environmental sampling as well. And so we developed a comprehensive training program around this that includes training on all aspects of One Health approach, so study design, sample collection, as well as the technical protocols, and of course the bioinformatics approaches that you need. And then providing exposure, importantly, and networking with other experts in the field and other scientists that have common interests. So the overarching goal here is really to enable the generation of data for more informed decision-making and to improve hypothesis generation to drive better research and clinical studies, which of course leads to more effective public health policy and better use of resources. Okay, so based on this training program, about 2 years ago, we decided to enter into a partnership with the Gates Foundation, and where we created an RFA for pilot studies using metagenomic sequencing to address different public health challenges. And we used their mechanism that's called the Grand Challenges Exploration Grants, or GCEs, I'll refer to them as. And the support that they received from the Gates Foundation allows these groups to come to San Francisco for training at the BioHub, and for them to purchase a small sequencer for their labs and other reagents that they'll need. In addition, at the BioHub, in addition to their intensive 2 to 3 week training that they undergo with us, we also provide resources for sequencing hundreds of their samples, as well as ongoing support to these groups as they move on back to their home countries to set up their pipelines in-house. And then through the Trans-Network Initiative, of course, they have access to the analytical pipeline IDSeq that I mentioned, as well as access to high performance computing in the cloud and free data storage. So all this really works together to establish, I think, a solid program for metagenomic sequencing that can then grow and expand beyond. This is also an extremely flexible platform. It's very adaptable to address a wide range of public health questions. So we were able in the spring of 2019 to award about 10 of these grants. And this is just a map showing you the locations of the labs that we currently work with. And this is also kind of a list of the different topics and questions that they have, from pathogen identification to vector surveillance to understanding causes of fever and neonatal mortality. So 6 out of the 10 groups were able to come to San Francisco and complete their training prior to the pandemic hitting. And 3 of the 6 that came were able to actually set up their metagenomics pipeline and get it up and running rather consistently before the pandemic struck. And we were poised to travel to start doing some site visits, but then all the borders closed, so we had to unfortunately put that on hold for a while. And so not all of the groups that we worked with had a chance to really set up their pipeline and in-house before they had to kind of pivot towards helping their country's response to COVID-19 and setting up testing there. So now I've told you a little bit about the program and how we thought it would work. And so now we're going to talk about, I'm going to share a few stories with you about some of the groups. So first up is our group in Madagascar. This is Team Fruit Bat. And they were really interested in zoonotic disease. And so we currently think that about 60 to 75% of emerging human infections are zoonotic. And so Kara's group did a study to really look at host and to delineate host and viral traits that were predictive of mortality, human mortality, associated with viral spillover. And they found some very interesting things in their study, and one of which was is represented here on the left, which shows that as you increase the phylogenetic distance of the host away from humans, which is represented on the x-axis here, and you compare that to the mortality rate, you actually find that the further away you are from humans, the more likely you are to cause significant mortality if there's a zoonotic event. And so highlighting here in the purple dots is the order that contains bats. And so not surprisingly, there are some notable examples of Marburg virus, Nipah, and SARS, of course, that we're all very familiar with. So this was really the jumping point for them to kind of explore what kind of emerging infections are happening in their bat population, and in addition to this type of work that they've done, there's been a lot of serological evidence, of course, of circulating antibodies to these types of viruses in Malagasy fruit bats. And to your right, I have some photos of 3 of the endemic species that are specific only to Madagascar of fruit bats. And these are the ones that the Madagascar group studies and catches to do some biological sampling. And so most bats on the island do come from either Africa, some of them do come from Asia, but these 3 are specific only to Madagascar, so it makes them really compelling to study as part of the zoonotic vector. There's also evidence of circulating antibodies specifically for Ebola in Malagasy bat hunters. And so despite kind of the bulk of the serological evidence for exposure, really they have yet to discover or to detect actual virus in either bats or humans for any of these that they have serological evidence for. So in order to get at this, they took advantage of 2 separate studies that were going on in the same region to perform some kind of preliminary metagenomic sequencing to understand what the pathogen landscape actually was in both bats and humans, and specifically humans with fevers of unknown origin. And so they took biosamples from wild-caught bats, and specifically the endemic species. And then they took a variety of samples from febrile patients that were undiagnosed. And so what I'm showing you here is actually a Kamer-based phylogenetic tree that was developed in IDC. And what they found was a lot of beta coronaviruses. And I want to remind you that this actually analysis was done in December and January, so right about the same time that things were happening in China, and we were just getting news of what was going on there. So this was really exciting to us, and this is just showing that whether you took nasal swabs from humans or feces from bats, we actually picked up a lot of different coronaviruses. And so really the extent of circulation of coronaviruses was really underappreciated in Madagascar. And so they thought that this really could be a potential for zoonotic events. And another point that I just want to make here for this is that the NP swabs really yielded some other viruses other than beta coronaviruses, but also yielded complete genomes of some of the viruses that they were looking at. So now if you build out the consensus sequence and rebuild the tree for beta coronaviruses, you can see here on the bottom the human coronavirus that they've pulled out of some of the samples, as well as a novel bat beta coronavirus that's pictured also in yellow. And so they actually found 2 novel covies in this specific subgenus. And so this was really exciting that they were able to do this and pull this out. So they're looking more and actually into this and exploring this more right now. In addition to this, they also, excitingly, were able to identify a novel sequence of a potentially zoonotic henipavirus in urine samples from these bats. And so this novel virus actually turns out to be very divergent from the other species that have currently been described in bats. And so here, again, for the first time in a Malagasy bat species, they were able to identify henipavirus. And so this, again, was really exciting that they were able to pull out so much great data from this first run. There were other paramyxos also identified in this group, but kind of coincidentally this week, there was just an article that came out about them finding Nipah virus actually in bats in Bangladesh. So again, another first that they've actually now, using these sequencing technologies, have been able to pull out and identify actual virus. So in summary, the metagenomic sequencing here really allowed them to discover both novel beta coronaviruses, as well as for the first time the existence of, demonstrating the existence of a henipavirus in a Malagasy bat species. And it's very clear that this is a demonstration of how metagenomics can be used as a surveillance strategy for pathogens with zoonotic outbreak potential. And these data now are providing an avenue of future collaboration between our Madagascar group and other, not just African countries, but also countries and groups in South Asia. So we're really excited about the work that they've been doing. The next group I want to talk about is from Cambodia. And this group was really interested in vector-borne pathogens and understanding what kind of exposures were happening in their region to help them define kind of biomonitoring strategies that they can use and an effective vector control efforts for their area. So Cambodia is one of the most rural countries still in Southeast Asia, but it is rapidly urbanizing. And so there's a lot of changing health needs that are occurring in the population there, and they really need to do more studies to address what they are. So they know that they actually have a lot of infectious disease. Dengue is known to be hyperendemic in Cambodia, so they assume they're going to see a lot of dengue from year to year. But despite the kind of the numerous types of infectious disease that do circulate, they seem to have a kind of a dearth of diagnostics that they can use to kind of get at the causes of a lot of illness there. And so that's why they turned to metagenomic sequencing. And in their pilot study, they decided to take a sampling of their community. And they took febrile patients and sequenced them. And about 70% of them so far have been sequenced. And almost half of them have gotten some positive pathogen hits. And so this is what the data looks like. And so this is just a representation of some of the different pathogen hits that they've got. All of these were collected over 2019 and 2020. And of course, the biggest thing to notice is not just that vector-borne disease really kind of comprises about 80% of all the pathogen hits here, but that in 2019, they had a huge dengue outbreak in the region. What was surprising, though, was that dengue 2 has always been the prominent serotype in Cambodia. And so this survey actually revealed that dengue 1 was also quite prominent this year. And so this allowed them to do some immediate genomic epidemiology. And they did this. This is another representation from IDSeq. Again, using a Camer-based approach to just look at what the nearest relative was for a lot of the strains that they were pulling out. So in addition to this, I'm going to talk about two more little stories of some of the findings in the data here, specifically about this one Zika hit that they got, as well as finding Plasmodium knowlesi in their samples. So here is, again, another visualization that you can get in IDSeq, and this is really showing you a config assembly of the Zika virus reads from the sample that they had in their cohort. And it's showing really nice coverage of the genome. So depending on the size, you can, with not too much sequencing, actually pull out a full genome in this case. And what they decided to do when they saw the Zika was to do a seroprevalence study on a subset of pediatric patients in their cohort. And what they found was that about 7% of the children that they surveyed had neutralizing antibodies to Zika virus. And so really, this preliminary data suggested that Zika is circulating at a much higher rate than they previously appreciated. And so that led to implementing basically expanded testing to evaluate flavivirus immunity and cross-reactivity, which will be really important prior to them implementing any kind of dengue vaccine in Southeast Asia. So next is the case of a forest worker who lived nearby the sampling area. So this is a map of showing you kind of the malaria incidence across Cambodia, the case rate. And although deaths from malaria have gone down significantly, as shown by the green line in this graph here, the number of cases actually hasn't really gone down all that much. And so they still have a significant amount of morbidity as a result of malaria. And about 99% of all cases of malaria are caused by either Plasmodium falciparum or Plasmodium vivax. And again, there's some very forested areas of Cambodia, and one of which that was close to the clinic that was doing sampling, again, had this patient come in. And this person actually was negative by malaria rapid test as well as PCR. And so what they did was they did metagenomic sequencing on the sample again and analyzed Bioseq to find that it was, in fact, a different strain than they were expecting, but Plasmodium knowlesi. And the interesting thing here is that this is the first diagnosis of Plasmodium knowlesi in over 10 years in the area. And so this was quite surprising and led them to implement the implementation of a PCR for knowlesi along with Plasmodium vivax and falciparum as a regular course now. So in summary for Cambodia, they really have shown with their work here that the pathogen landscape was much more diverse than they had anticipated. And this is really a nice example of how biomonitoring of vector-borne disease and prediction of it can be successful through the combination of both metagenomic sequencing and seroepidemiology. And finally, this success has led the Ministry of Health to request an expansion of the use of both sequencing and the IDSeq software into 2 pediatric hospitals in Phnom Penh. So I mentioned to you before about the pandemic. This is all at the forefront of everyone's minds, obviously. And I mentioned that we had to put some of the in-person training with our groups on hold as a result of this. And this is a map of what things looked like in February when we decided to put things on hold and when borders were shutting down in many areas. But what actually we realized looking at this map was the fact that a lot of the groups that we work with were actually on the front lines of this outbreak. And so we realized very quickly that we needed to help them start setting up sequencing for themselves for specifically for SARS-CoV-2. And so amidst of all the testing and helping out of the DPHs that we were doing here in California, we got back in touch with our groups, and in fact, they were quite successful in establishing sequencing for SARS-CoV-2 in their countries. So just to mention a few of them, this is a next strain visualization for the strains currently that have been coming out of Africa. And this is just to really highlight the fact that in Africa, the vast majority of the SARS-CoV-2 sequences are being generated by a really small number of groups. And this just shows some of the countries that have contributed the majority of those strains. But here is Madagascar, and this is our group that contributed to, and 2 of the only actually SARS-CoV-2 genomes right now that exist on NextStrain. But they were really pleased to be able to do that themselves. This is a map of the Asian side of things with SARS-CoV-2, again, a NextStrain phylogeny. And here, we can see that our Cambodian partners were one of the first GCE groups actually to identify a positive case. It was from a traveler, and they sequenced the full genome and were able to upload it to GISAID also using an MSSPE method that was developed in collaboration with a very talented scientist in my group, Vita Ayang. And they needed to really do this in order to enable to get full coverage of the genome using an ISeq 100. Another group that we work with in Bangladesh at the Child Health Research Foundation also made history by being the first in-country lab to sequence their own SARS-CoV-2 genomes and upload them to a public database. And so this I'm showing you here, the one was the first sequence to go up from Bangladesh, and the the storm that it happened afterwards, they got so much attention for having done this that it really set off kind of a domino effect of sharing protocols with other institutions and getting other groups to also sequence and contribute genomes to the database. And so that's what's circled up top there. So this was really exciting for us, and we're really happy to see the successes of our groups in doing this. But how, and I hope that I've been able to illustrate kind of some concrete examples of how metagenomic sequencing pairs easily with accessible, cloud-based analysis pipelines, and how it can be really impactful and an agile platform really to assess One Health questions in public health and for disease outbreaks. So now, though, how do we think about and how do we measure success for these groups? What are the metrics beyond just creating a library and sequencing it that we can use? And so I kind of think about it with three major areas. One is contributing to the global research community. And so all of these groups that we've worked with so far have demonstrated being able to publish genomes. They've all given presentations at different international conferences, as well as written manuscripts. And what all this does is really increase recognition, not only in their own countries, and by their ministries of health and other organizations they can potentially work with, but within the greater research community as well. And this really makes it easier to make connections and to find collaborators. Secondly is being able to build a sustainable capability to do sequencing locally. And so this really has been done because they've trained other personnel, so they've grown their workforce, so to speak. So training the trainers was really our goal, and a lot of the groups have shown to be able to do that. Securing additional funding from sources beyond the Gates Foundation has been key for a lot of groups. And also, they've shown that they can build both intra and inter-organizational networks. And so all of these things really create a more dependable pool of resources to draw from and a stable set of resources for the group. And finally, the ultimate goal here, right, is to inform public health policy. And so these groups have been examples of how they've been able to identify the need for new diagnostics, the need for new surveillance initiatives that they've implemented, and of course, for doing outbreak investigations. And this really gives you, puts evidence in their hands to make decisions that will ultimately benefit the health of the community and the country for better health outcomes. So in thinking about this, we've really understood kind of through our experience now over the last 18 months or so that this type of program takes contributions from both the trainers and the trainees to really make it successful. And we've been able to identify a number of things that can both promote and hinder this type of success. And so on the BioHub side, we really do this in a number of ways. We've really tried to reinforce both the didactic and practical training that they get through site visits, being there in person with them side by side, and creating opportunities for them to exchange ideas and solutions with other people and other groups that are setting up these platforms in their labs. So really creating a community of sequencing labs. We provide access to tools and software, and not just that, but also to personnel who are experts in the field that they can tap into. Again, this is a true partnership between the BioHub and our partner labs. So we are side by side with them, giving them hands-on training, protocol testing, and really troubleshooting things for that are tailored to the individual needs of the labs that we work with. There is a very big investment of time, and this isn't a one-day course or a one-week course. This is, in fact, many months of work that we invest to get, to stay with these groups and take them through step-by-step and through a lot of communication and being readily accessible to them all the time. And finally, we really focus on mentoring to truly develop strategies for sustainability for these different groups. Now, on our partner lab side, they have a number of things that we've seen that have made them really successful. So groups that have leadership that is engaged and that prioritize setting up sequencing in their labs tend to be much more successful. And communication, I can't stress this enough, that especially now when we're doing remote training for the groups that haven't been able to come to work in the lab with us at Biohub. Communication is really key for making this successful. Having dedicated personnel that are responsible for setting up the platform in country is really important. And establishing that chain of training, so doing the secondary trainings beyond the group that we train at Biohub is super important for keeping this sustainable. And finally, groups that can advocate for themselves. So cultivating that interorganizational relationships that they have and to help make their public health endeavors much more productive going forward. So we've really found this to be a very enriching process, and we're really excited to continue working with our groups. We will, of course, continue on with all of the groups remotely for some of them, unfortunately. But these are some pictures from some of our GCE groups. And of course, we've had a very fruitful and fun collaboration with Wandwasan and the Global One Health Initiative at Ohio State, where we help train their East African Fogarty Fellows that come and visit to Ohio. And we've also engaged into a partnership with the Africa CDC, where we're training various sites for sequencing on the continent. So we're really excited about that partnership too, and hope that continues into the future. And so with that, I just want to acknowledge kind of all the people that have worked so hard to make this successful, both the Biohub and the Chan Zuckerberg Initiative, as well as the Gates Foundation to help make this possible. And of course, I just want to acknowledge all the hard work that's been done by the groups whose stories I shared with you today. And so with that, I can take any questions.
Great. Thank you so much, Dr. Chato, for a really fascinating talk and summary of some of the great projects you have. We can take time for some questions now before we go into our other plenary or our other panelist discussions. So we have one question that's come in from Christina Petten. Do you have any updates to share from the collaboration and training with Brazil partner groups? Thank you, very fascinating. Yeah,
Brazil was one of the groups that unfortunately wasn't able to make it out to San Francisco before the pandemic hit. And so there were about 4 of these groups, and all of the groups actually, as well as Brazil, had to really pivot again, as I said, to addressing the needs of their country for COVID-19. And so Brazil has been really wrapped up in doing that. But I was just on the phone with them yesterday and talking about how we're going to start up doing remote training with them and how we're going to move forward. So they're just starting out on setting up sequencing. They have nanopore sequencing that they are able to do, but the Illumina sequencing, the short read sequencing, for metagenomics is what we're going to embark on next. So they're just starting out kind of on that, but we're really excited to work with them. They're in Salvador actually, and it's a fun place to visit. So I'm looking forward to going back.
We're all looking forward to being able to travel.
Yeah, exactly.
I have a question. Just moving forward, we're at a really odd time, and there are just, having watched your presentation, I'm really excited about potential collaboration with the Biohub. It just looks wonderful. But are there new models emerging here with your Biohub that will allow for the initiation of new collaborations, even though we can't all get together in person, but perhaps virtually? For example, we have a big interest in Epstein-Barr viral genome variability across the globe. Our early studies in South and Central America compared to North America, and the published genomes in Asia are incredibly varied. And this has huge implications for vaccine, and we'd like to move in that direction, but don't know if that's a possibility at this point, given our current challenges with COVID.
Right. Yeah, I agree. It definitely has made things harder when you can't interact with the world, but you have to do everything from your own office or your own lab. But we are actually always developing kind of new tools and ways to data share. That's actually been quite a focus of the BioHub is how do we make data more accessible to people to use it where they are in their labs, and for sharing that, as well as we're in the process always of thinking about new tools that can be used, new diagnostics that can be used and implemented in low resource settings. So there's always new stuff kind of brewing, and I didn't have time to talk about it today, but there were a whole number of initiatives that kind of we undertook to address SARS-CoV-2 and address kind of the shortfalls in respirators and things like that have occurred here in the U.S. And so, I think there's almost too much going on for me to be able to explain or even know about. And it's just great to, if you have an idea, just engage us and talk to the folks there, because we're always interested in new ideas and talking to new people, because who knows, there might be, some new technology or something new that we've been working on that might be a perfect fit for what you need.
That sounds great. Thank you. We have another question from Sally Miller. There's a great need for identification and surveillance of plant pathogens that affect food security. Do you anticipate expanding your collaborations to plant pathogens?
Yeah, this is so easily done. I mean, as long as something has DNA or RNA, you can sequence it, right? So this is very, very applicable for doing that type of, for those types of questions and doing that type of surveillance. We haven't had, that hasn't been the forefront. We focused on infectious disease, but we are certainly open to talking to groups that are interested in doing that, and would love to help them get that up and running.
Okay, and then we have another question from Rafael Vieira. Any data on microbiome in ticks from Africa? We're performing a great study in Somalia and UFPR.
Wow, that's really cool. So not in Africa, but actually one of the investigators, the same one who works with our Madagascar group, Amy Kistler, has been doing a tick project here in California, actually with some investigators at Berkeley. And so I think they would be super interested in talking to you and collaborating on something like that.
Great. Any additional questions? If not, we can move on to our next speaker. Our next speaker is Rosemary Rochford from the University of Colorado. Dr. Rochford's research has been on endemic Burkitt's lymphoma, the most common childhood cancer in sub-Saharan Africa. Endemic Burkitt's lymphoma is linked to infection with both EBV and Plasmodium falciparum, malaria. Through her research program based in Kisumu, Kenya, she's uncovered a number of ways that repeated infection with Plasmodium, malaria, dysregulates EBV persistence and ultimately increases the risk for Burkitt's in children. More recently, she's extended her studies to Uganda to understand how malaria increases susceptibility to Kaposi sarcoma herpes virus, the causative agent in Kaposi sarcoma, otherwise known as HHV8. We collaborate directly with Rosemary on the malaria and EBV project, and it's a pleasure to introduce her to give her first talk. Thank you, Rosemary.
Great. Okay, so I'm going to sort of switch from infectious disease, and a lot of what Christina was talking about was more RNA viruses, and I'm going to talk about the virus, my favorite virus, EBV. And this virus is the cause of infectious mononucleosis, and it's a very successful virus. In fact, I'd say it's the most successful. 95% of the world population are seropositive. Once you're infected for life. When you look in the peripheral blood, it's found in the B cells in healthy people, but a very low frequency, higher in Africa than in the U.S. populations. And it makes a challenge for sequencing that way because it's hard to pull out that rare population. We know that there's an estimated 200,000 cancers each year linked to EBV worldwide. So it's a real, from the cancer point of view, it's an important problem. And I want to sort of bring up a couple of things about EBV that thinking about sequencing and genomics. So this virus was the first virus found to be associated with human cancers. It was identified back in 1964 by Tony Epstein in collaboration with Dennis Burkitt. And this is an example of a north-south collaboration that began many, many years ago, obviously. I always talk about the story of the curious scientist in a great lecture where Tony Epstein was at a lecture that Dennis Burkitt was talking about this cancer in these children and was able to get these samples shipped up from Uganda to England and identified the virus by electron microscopy. So that was that's why I love this picture here, the beautiful EBV-EM picture. It was the first large DNA virus sequenced. It was sequenced in 1984. So we've had the genetic information for a long time. And now the question is, how do you bring that information to understand the pathogenesis of disease? And one thing I would say that I'm when we think about sort of the One Health and global health is that a lot of work on EBV sort of moved to the laboratories and studied it in the molecular biology, and there's a lot of great information there. But you actually have to bring it back to where the virus is causing disease and understand that interaction. And so that's what I'm talking about today. This picture, this is one of the things EBV, just to give you an identification of it. And if you'll notice, it's a fairly large genome, 172,000 base pairs. And again, gives you a great appreciation for what they did back in 1984 with Sanger sequencing. The virus was digested with a BAM-H1 restriction enzyme and made into 26 fragments, which is why we have these BAM-H1, B, K, R, Z. So the gene I'm going to talk about is BZLF1, which was the BAM-H1Z fragment left with reading frame 1, so that's where the name came from. And what we know is that we've done a lot of work on the latent phase of the virus, and people have studied the latent genes, the Epstein-Barr nuclear antigens, and the latent membrane proteins and their role in oncogenesis. But there's a lot of information coming out that maybe the lytic cycle might be more interesting, and that's what I'm going to talk to you about today. So it's not just about making more virus, but might be linked to cancer emergence. And so we're going to talk about BZLF1. It encodes the Z protein, also known as Z transcription activator, or zebra. And this protein is necessary and is sufficient for induction of the lytic cycle. So when it goes from a latent to lytic phase, what we'll see here is that you're going to get the latent viral genome. You're going to get the immediate early, which is the BZLF1 and also BRLF1 transcribed, and then the proteins, you have early and late, and then progeny virus. So in 2017, I just lost a couple decades there. Paul Farrell and Paul Kellum used next generation sequencing, collected samples from around the world to look at, they did 241 EBV genomes. So just to give you a framework, 1984, the first EBV was sequenced. The next sequence of a full genome was in 2007, and then not till 2017 that we have this sort of larger composite. And this is a principal component analysis, just showing you that there's actually these two strains of EBV, EBV type 1 and EBV type 2. EBV type 1 is the one that's found around the world. EBV type 2 has much more restricted variation. And a lot of the variation is in the EBNA2 and 3A genes, and this is just shown here. These are these latent genes that are known oncogenes. But when you look at the sequence, and I'll just say one of the things when you're looking at large genome sequences, and you try to do this meta-analysis, it didn't pull out these, we're going to talk about differences in BZLF1, and also we found differences early on in GP350 and GP42 when you just pull out those sequences and look at them. And so it's a different way of thinking about it. So you have the metagenomic comparison, but you also need to focus in on the individual genes. So main differences in latent genes, but there are also differences in lytic. So one of the things that this was work done and had been seen before, but there's the Z promoter that drives that BZLF1, which is the key transcriptional transactivator, and the prototype promoter ZP-P, but there's a variant promoter ZPV3, and it just has a single nucleotide difference, creates an NFAT C1 binding motif, confers greater enhanced lytic replication when you have that ZPV3. It responds more strongly to the B cell receptor stimulation of B cells, so it induces to the lytic cycle much more easily. And it's found in all the EBV type 2 strains, but it's variable in EBV type 1. And in fact, when they looked at those sequences in that paper by Correa et al., most of the strains had the EBV type 1 had that the prototype promoter, but a subset had that B3 promoter that enhanced lytic replication. And again, the EBV type 2, all of them have that. So the work that I've been doing has been at the Center for Global Health Research in Kenya Medical Research Institute in Kisumu, Kenya. And it's a beautiful site there. And this is a picture of my now former PhD student, Ibrahim Daoud, who's actually doing a lot of work with HIV surveillance, director of the laboratory research for Walter Reed now. And within there, we've been looking, I've been actually as a postdoc, I was interested in this difference between, you have this common EBV type 1 strain and then this rare EBV type 2, and there was a few papers published. So we looked at samples we'd collected in our study and looked at what the frequency of type 1 or type 2 was in the population. And what we saw is that in both the blood, peripheral blood, where the virus is latent and also the virus is shed in the saliva, you can see a lot of co-infection with type 1 and type 2 viruses, quite a bit actually. And we have another paper showing that infection with one virus doesn't protect against infection with the second virus. So there's something going on in terms of that and has implications for vaccine. But the fact that this type 2 was also common has led us to a lot of other questions in terms of what is type 2 doing. And just as a segue, we've actually found that EBV type 2 infects T cells as well as B cells through genetic variation, we think, of the GP350. So again, this sort of brings this idea that if we look at, you can have the whole metagenome and a comparison of sequences, but focusing in on the individual genes and trying to understand that genetic variation helps us begin to understand a little bit about differences in pathogenesis. So I'm bringing up a study that we've done. It was a longitudinal birth cohort study to look at whether maternal exposure to malaria influenced the age of EBV infection. And the point I wanted to make is we followed these mothers. We enrolled them at their first A&C clinic, looked at infection with EBV and malaria, HIV, and other worms and different infections. And then once post-delivery, we looked at breast milk and saliva in these mothers, and then we followed the infants up through 24 months to really understand how that maternal environment influenced susceptibility to infection of EBV. So what we found just in looking at that, we enrolled in the first sort of half of the study, 200 pregnant women, excluded the HIV positive mothers, and found that the EBV load was higher in women that were infected with malaria. So there's this dynamic interaction of malaria and inducing EBV reactivation, we think. And if there was more malaria, the viral load increased as well. But one of the things we wanted to look at is if we had found that kids were infected with, subset were infected with EBV before 6 months of age, which is not what we see in the U.S. population. Here in the U.S., typically you'll see a subset of kids infected about 2 to 3 years of age, and then there's a delay of infection until teenage years, and that's where you get infectious mononucleosis. In the Kenyan setting, we see that the children are infected, almost all of them are infected with EBV by 2 years of age, and a subset were infected at less than 6 months, and that's sort of what made us think about what's going on with the mom. We decided to look in the breast milk because at before 6 months of age, 2, 3, 4 months of age, you don't really see a lot of saliva transmission potentially. And what we found is that actually at 6, 10, 14, 18 weeks postpartum, you could find EBV in the breast milk, looking at by EBV in the breast milk, and higher viral loads were associated with mothers who had malaria at delivery. Now, one of the important things, though, is you can detect a virus by PCR, but you want to also look for whether the virus is infectious. And so we did a number of experiments, but importantly, what we found is that if we took those breast milk samples and incubated them with peripheral blood mononuclear cells, we resulted in generation of EBV transformed lymphoblastoid cell lines. This is just showing you the clumped cell lines just infected directly with the breast milk samples. So the mothers are excreting infectious virus in the breast milk, which is really interesting. So we generated these LCLs, and then we set up a collaboration with Shannon Kenny at University of Wisconsin, because she was very interested in this variation in the Z promoter. And through this collaboration, we showed that if we look at the Z promoter, the ZPV3 variant, that one that it causes more lytic reactivation, has that NFAT binding site, is not found in the peripheral blood. The prototype promoter is, but in these samples from the breast milk, they were all EBV type 1, and all of them had the ZPV3. So that is overrepresented in the breast milk samples in these mothers that had malaria during pregnancy. So it suggests that we're getting sort of a driving towards with that variant in the type 1 virus, allowing them to be more lytic and more infectious. And then again with Shannon Kenny, trying to look at this, the frequency of both EBV type 1 and type 2 in Burkitt lymphoma, just to show that in the Burkitt lymphomas, you tend to have more type 1 EBV than type 2. And there's a paper that just came out that also confirms that using next generation sequencing. But in addition, if you look at that ZPV3 variant of the type 1s, it's overrepresented in the Burkitt lymphoma. So it suggests that there's something about that ZPV3 in the type 1 virus enhances their ability to be more infectious. And I just want to bring it back to sort of from the genome side of the study. We had published this paper back in 2010, looking at children with Burkitt lymphoma and seeing what types of antibodies they have. And you see an elevated antibody titer to the antibody, to ZTA, the BZLF1 protein. And so they also had higher antibody titers to VCA, and that's been well known, but the ZTA was fairly striking and significant versus the EBNA1 IgG. So there might be something in terms of the pathogenesis linked to that reactivation. And so the question is we have two ideas. One is the dysregulation of EBV persistence, so that what we hypothesize is that malaria is driving more EBV reactivation. There's more virus. That virus then infects more latently infected cells. That increases the risk for cancer within that. And if there's a promoter variant that tends to be more susceptible to that, then that becomes interesting to understand its link to increasing risk for cancer. And just to sort of wrap up here, this is a paper that just came out in Cancers, and thinking about, rather than we think about, I've been focused on sort of the lytic cycle and the virus. If there's more virus, it infects more cells and what goes on. But they actually have a hypothesis that you can have an abortive lytic infection and creates maybe an environment that's tumor promoting in and of itself, so that you have something that triggers that BZLF1, the protein gets made, and that creates genomic instability, and it's been shown to decrease P53, increase BCL2, which would prevent apoptosis, and create an inflammatory environment, and so on. So I guess what I want to try to say here then is just to think about integrating it so that we have a large virus that's been sequenced in 1984. We now begin to see the compendium of more sequences, and again with Rob talking about throughout across the world and looking at those sequences, and then bringing that data back to the pathogenesis of the cancer. So I'll just stop there. And acknowledge the group that in Kenya in particular, I've had the pleasure to train a number of graduate students there. These are all my former trainees, and then Emily is going to be a new PhD student, the work in University of Colorado as well. And I show this picture. These are all children with Burkitt lymphoma, and my husband, that's his very expensive $150 sunglasses. They were having a great giggle putting them on, but that's the cost of treatment in Kenya for Burkitt lymphoma. So to remind ourselves that we really need to understand, to take the understanding of this genetic variation and how do we ultimately prevent this cancer. So I'll stop there. Thank you.
Thank you, Rosemary, for a wonderful talk. We're going to move on to the next panelist, and then we'll have a session for question answer following our third panelist discussion. So for our next speaker is Zelalem Makaria from the Ohio State University College of Veterinary Medicine. He is the Associate Director for the Infectious Disease Molecular Epidemiology Lab at OSU and works closely with the Global One Health Initiative. In his role, he oversees the laboratory activities for GOHI or Global One and coordinates training for postdoctoral and PhD fellows in the area of molecular diagnostics, genomics, and virology. Dr. Zelalem has a PhD in molecular virology with additional training in the areas of applied genomics and bioinformatics. He's been a postdoctoral scholar at the Maxwell Gluck Equine Research Center at the University of Kentucky, where he extensively used next-generation sequencing and technologies to investigate evolution and transmission dynamics of rapidly mutating RNA viruses. He also worked at Louisiana State University, where he was involved with the establishment of new laboratory with the aim of advancing next-generation sequencing methods for diagnosing for diagnostic applications. Today, Dr. Makaria will be speaking on the applications of single molecule sequencing for rapid identification of microbial pathogen in the clinical setting. So with that, I'll hand the screen over to Zelalem. Thank you.
Thank you very much, Dr. Bioki. It's a pleasure to be in this panel. As said, I will be talking about a single molecule sequencing today, also called third generation sequencing. This is the work that I have been involved while I was a postdoctoral scholar in Louisiana State University in Louisiana Animal Diagnostic Laboratory. But as an introduction to show you how I end up using this third generation sequencing or single molecule sequencing, I'll just go back a little bit and start from my work at the University of Kentucky, which primarily involves around, characterizing equine arthritis virus. A pathogen only infect equids, doesn't have any, potential zoonotic activity, but causes significant health and economic repercussions. So my primary role was to study the evolution, the intra-host evolution of this particular virus in stallion reproductive tract. So one thing about this particular virus is it forms a carrier state or persistency. So the persistency only forms in stallions. It doesn't form in geldings or foals or in mares. It just only persists in stallions, and it can persist for years or for lifelong. So we had a very good model in University of Kentucky to study this persistency, especially the evolution of the virus during persistent infection. So we had naturally infected horses that were shedding this virus for more than 10 years. And the samples were, kept regularly. And then we have those samples to characterize the infection for 10 years. And we have also done experimental infections of horses, which goes for 2 years, and collected samples longitudinally for 2 years. And we did some deep sequencing to characterize the change of these viruses through time. And we found out genes and mutations that are associated with this persistency. So that work, while doing that work, the most convenient and the industry standard way of characterizing this intra-host variation within the RNA viruses was the Illumina sequencing platform. So we utilized that sequencing platform to characterize, yesterday, Dr. Saif was talking about quasi-species. This is the quasi-species population that we characterize in those experimentally infected stallions. So after we do that, we have come up with a very good picture of how that virus evolved. And we combine that data also with whatever available in the gene bank and also with some archival data and get a very clear picture of what the evolution of this virus seems since its emergence. But one thing that was remaining was if we want to understand recombination or group selection, how these mutations connected to each other, whether these mutations are occurring in a single strand of RNA molecule or a DNA in virus population, the Illumina sequencing platform is not going to be a big hub. Because of its short read, we need a long read. So I start researching about the technology that can overcome this obstacle, and then find out about these third generation sequencing instruments, particularly the nanopore mini ion. In the meantime, our lab also from University of Kentucky moved to Louisiana State University and then situated in diagnostic inside the diagnostic lab. So we took our mini ion with us. So there, the only sequencing instrument that was available for me was the nanopore platform, but there was also in the core Illumina sequencing. So we had a chance to play around with both. But today I will be focusing on the nanopore technology. So these nanopore and packed biotechnology, which we call them a single molecule sequencing technologies or a third generation sequencing technologies, have particular advantages. They have also disadvantages. So as I already said, we use these second generation platforms to get in-depth look of this viral population. But if we look at resolving the genomes very easily, completing the genomes in less computational way, then the nanopore technologies become very important ones. So since this is a technology relatively came about to the world of sequencing very recently, let me take you through the anatomy of this instrument. So this is the mini ion, the instrument which I extensively use to do some bacterial sequencing as well as metagenomics studies. But today I will be focusing on the bacterial sequencing. And then it has a sequencing chip, a very small sequencing chip that would be inserted into the sequencing device. So within that sequencing chip, you will have such kinds of anatomy, which is a nanopore and a protein. The nanopore are bound to a membrane, and then the company have a patented protein, which they call it a motor protein, that will bind to a specifically prepared nucleic acid and strip it through the nanopore, as you can see it in this animation. While that happens, there will be a fluctuation in current, and that current will be captured in a native file format called FAST5. That FAST5 file can be converted into what we know a FASTQ file and can be analyzed as a normal sequencing. So it's a nanopore bound with a protein, and then you have to prepare it in a certain way and flows through. So this is a very a disruptive kind of technology, because most of the previous technologies use polymerases or amplification system to sequence. This is just directly attaching the DNA into a motor protein and pass it through a sensing device. So there are many advantages of this technology that caught us to, caught our attention. And even after I come to the Global One Health Initiative, we're really interested in this device because of several merits it has, especially in the context of, developing countries. Even with the current ongoing pandemic in COVID-19, Dr. Tato earlier mentioned that there are several genomes from Africa. 20% of those genomes are currently being sequenced using these instruments. So there are particular advantages that switch it to such use. So one of the most important thing is the read lengths. As I said, it is a long read sequencing technology. So you determine, the laboratory person who prepared the library determine the read lengths. If carefully prepared library can give a read as long as the DNA or the RNA is not broken, and properly prepared, attach all the adapters and process it through the flow cell, you will get the entire DNA or RNA being sequenced. The other thing is real-time analysis, which I will try to touch base and show you some data as how you can employ the real-time analysis to make immediate clinical decisions and then go on. It's a portable sequencer. I've seen yesterday, some groups use a battery pack to fully sequence organisms from remote areas. And it can be also scaled up and scaled down. It can be, very minimalistic, which I will show you our experience. And then it can be also, you can, a laboratory or an institute can purchase a very high throughput machine to scale up this kind of sequencing analysis. So for example, this is the setup I had. So we just got like a very small grant from AVLD and the Thermo Fisher, a diagnostic innovation grant. So we spent all the money on flow cells. Here are the list of flow cells which I purchased with that grant. So because it was a diagnostic lab, three things were important besides the regulations and the other things. Accuracy, you have to properly type the organisms that's coming. Speed, because the clinicians, it's a veterinary diagnostic lab, so it's associated with the clinic, and then the clinician require the result in a really reasonable amount of time, and cost. So cost, when I say cost, in terms of running cost as well as infrastructure cost. So for example, this is the setup I had in Louisiana Diagnostic Lab. We had 3 mini sequencing devices connected to a GPU unit with nanopore cells. So that's like a small powerful computer that can attach to the sequencing device. But we spent most of the money we got on for data analysis buying a very good Mac computer. And I used to call that particular instrument GridION X3, which Nanopore doesn't know, because they have a GridION 5 machine, which they have a 5 flow cell and a GPU device in a box. So just to compare it with that, this is like a $50,000 initial cost. This is, we just spent around $10,000 to set up this. So this was a very, very small experiment we did during my 1-year stay in Louisiana State University. So we purchased all the flow cells and then arranged them to come in a different time in order to keep the expiry date very current. And then we intervene on a certain workflow. So the particular workflow which I would like to talk about today is the bacteriology workflow, which I came to understand while I go to the Louisiana State University. So what they used to work is the samples will be submitted, so the regular culture will be done. Then once bacterial isolate pop up in the culture plate, they will do MALDI-TOF. But when speaking with the molecular section heads, what I understood is, when they do MALDI-TOFs, so because it's a veterinary diagnostic lab, several exotic animals present and several new pathogens come in. So sometimes MALDI-TOF might not have in a database the particular profile related to the isolates. So it will be unassigned. It will not be identified. So we thought of, if you can do quickly sequence using a nanopore sequencer, we can actually customize the MALDI-TOF database so that the next time when the same organism comes, it will, so basically it will help us to develop a custom database that can integrate into the laboratory system. So most of the work, the laboratory work, were optimizations of the nucleic acid preparation methods for different bacterials. So we tried like beta type or Colomanus type, things like that. And library preparation methods, we try to optimize it for yield. So the most important thing is adapter ligations. We found that increasing the incubation time rather than increasing the concentration gives a better yield. And also for low concentration, we tried read size selections by using Covartis tube, but the problem we get is it will automatically inflate the cost. So that, but generally we just develop a protocol within like 7 hours, 8 hours, that we can tell the molecular diagnostic section and the bacteriology section that this is the organism. You can use it for customizing your MALDI-TOF, or we can compare it with the other laboratory methods. So the bioinformatic workflow we utilized was one, we called it a mini flow. So we sequence, then we acquire the data in real time, and then we use the metric course or the nanopore system to send it to the cloud, and there will be what is in my port analysis, which they call it WIMP, and then we do that, and then it will automatically generate taxonomic IDs, and then that result will come back to our computer, and then we use that result to compare it to a more rigorous bioinformatic workflow. So it's illustrated here. So finally, once we do early inference of what that organism is, then we just go through a rigorous bioinformatic flows, which are customized for nanopore, and then compare them that we get it right. As key important things which I learned, especially working in diagnostic settings, is that quality control is so important, and especially nanopore as such a smaller company, there are several irregularities that you will encounter while working with them. So, for example, they require you to do an in-house QC, quality control. So this is the result of my in-house QC. So what you will look into doing in-house QC is the number of active pores in your flow cells, which means the green ones that you see on these pictures are active pores, which are ready for sequencing. But this particular flow cells is not really good because it has 215 pores. In nanopore, you have to get at least 800 and above. And so looking at the total numbers we get at the range of 200 to 1600, so 2 of the flow cells we get out of 20 has failed flow cells. So we have to return it and get a new back. So if you think this in a resource limited setting in Africa, so out of 20 flow cells, we get like 2 flow cells failing quality tests. So you need to have those consideration in mind in order to do this kind of analysis. Then we look at the distribution of active pores, and then there is what we call it a mask scan as well. When you actually run the sequencing, you can compare the quality control you did with the actual run, and then if the differences are more than 10%, then you can stop and investigate the run. But generally, most of the flow cells pass QC. I would say 10%. I don't know if from 20 making such assertion is good, but at least we get 2 flow cells not working the way it is intended to be. Then we run these sequencing experiments and assess the qualities. So in terms of looking at the quality, we see the yield and the individual sequence quality as well. So it's that small device you saw that can be on a palm of your hand can generate from 10 gigabytes to 25 gigabytes of data, and anywhere in between. This is a significant amount of data. Just to give you a perspective, this is the capacity of a MySeq machine in case of Illumina. And then that could range from 2,500,000 reads to 6,500,000 reads in my experiments. When we do a cDNA, we get a lesser yields. I heard very recently Nanopore has a new kits that can improve these particular aspects, but we generally got from 1.8 to 3 gigabytes of yield in case of a cDNA. Then I will assess the qualities, the read distributions. So if those of you who work with Illumina, you know the Illumina has a very high Phred score, but in Nanopore you don't get that. That's why you have to introduce a lot of post-run quality filtering steps. So our cutoff would be around 7, and then the median quality will be around Q10. Then we evaluate the pattern of data acquisition from all these flow cells we collected in 8 months' time. What we immediately noticed is the data acquisition is very quick. The moment you start run, you start acquiring data, and it quickly generates significant amount of data that can be utilized in the real time without waiting for the entire process to stop, or waiting for the machine to stop. You can actually acquire the data. So for example, as you can see here, even the first 5 hours, I was able to accumulate anywhere from 500,000 to 1.5 million reads. So our optimized protocol usually utilizes 10 bacterial samples to load it into a single flow cell. So if you divide this number by 10, anywhere from 50,000 to 100,000, more than 100,000 reads per bacteria can be get in the first 5 hours of this sequencing. We have customized our protocol to visualize this conduct or this sequencing in real time, so you can actually make a visual assessment. This is when the sequencing happening. So this is the first 5 hours of run. So you can see sometimes the size of the dots, long reads can come, and then I can actually look into the individual sequences here so that I can go into those sequences, take out, blast them, and identify what those organisms are. And so immediately also we can, categorize them into our different barcodes and make some simple analysis in how much sequences we get in 5 hours and what are the read lengths, and then use that information to make decision whether to continue or to stop, or to decide whether you have enough data to do your immediate clinical decision makings. So once we get this 5 hours data, so that the beauty of a nanopore system is you can actually program the machine to give you outputs in in 1 hour or 2 hours regularly. So we did a quick analysis of what is in my code, like I told you. Then you can see, like, we can profile these bacteria, some of them which like, for example, if you see these 2 bacteria, that probably doesn't have much information in genomic databases. You don't get that much hit, but for most bacteria, you'll get significant hits, so that the reads will be classified immediately into taxonomic groups. For most of the reference organisms that we use, even within the single hour sequencing time, we can classify them very confidently to what they are, to the species level. Then once you do that, this is an example of like Mycoplasma canis, we sequence it. Once you do that, you can actually gather this information later, and then work through the bioinformatic pipelines you have, and then come up with the full genome. So for example, one thing we noticed is a genome as like Mycoplasma, relatively smaller genome. Within the first 5 hours of sequencing, we can actually recover the entire genome. So this is like the plasmid using the de novo assembly graph. So just to give you a perspective, this kind of graph, if it were a Lumina reads, short reads, it will have such kind of graph structures, which the assemblers try to find the nodes and try to put it together. But for this one, it's very quick, and then we can assemble the plasmid very quickly. So also the coverage, as you see, very deep coverage, and then you can error correct the data and come up with your beautiful genome very easily. Thank you very much.
Very good. Thank you so much, Zelalem, for a very informative and interesting talk. Our last speaker is Anne-Catherine Eisfeld, who is a assistant professor at Ohio State. Dr. Eisfeld, her lab focuses primarily on the understanding of genomic underpinnings of acute myeloid leukemia. She was trained by Doctors Clara Bloomfield and Albert de la Chapelle, 2 highly distinguished and world-renowned experts in this field. Her initial translational research focused on prognostically significant molecular markers in AML and basic molecular biology and cancer genetics. In large-scale sequencing studies, Dr. Eisfeld revealed that molecular diversity of adult AML patients, including gene mutations and chromosomal abnormalities, and revealed important examples of how the presence or absence of those findings may influence treatment response and survival of patients with acute myeloid leukemia. Our current projects are focused on the clinical, genomic, and transcriptomic characteristics of myeloid sarcomas to identify better therapy and therapeutic options, as well as the role that novel cyclin gene mutations in AML play in leukemogenesis. The title of AK's talk today is Cancer Genomics for Everyone, and I will transfer the screen over to Dr. Eisfeld. Thank you.
Thank you so much. While I'm sharing my screen, I'd like to thank all of you for being here. This has been great so far. Thank you so much for giving me the opportunity to speak here today. And yes, the title of the talk was indeed Cancer Genomics for Everyone, but I put a question mark at the end because I think, should it be for everyone? And if so, how can we get it there? As I'm looking forward to the discussion after as well. Since the topic, like all the talks I hear, are so diverse, I decided to go a little bit broader and try to get really different aspects into my presentation today. I thought I'd start with my favorite disease, as Dr. Burkitt said, it's acute myeloid leukemia, and I thought this is a great example on how genomics transform a disease classification, also the treatment. And then I move a little bit, I still stay in AML, but also wanted to see what different testing scales are available, like from where we integrated genomic profiling, what the approaches are. And then the last thing I would just call location matters, share a couple of our thoughts from here, projects that were taken here in Ohio, and where I hope that could be expanded, how to enable genomic testing for everyone, or at least for a broader part of the population. So cancer genomics, I thought when I saw the header of the session, I thought it's very broad, and where should I start? And especially since we talk about so many other things, I think cancer genomics is an entire cosmos. Thinking, and this is going to be the main part of my talk, thinking about gene mutations, but really having to consider hereditary and somatic changes, thinking about chromosome changes that also go into it, not only numerical, but also copy number variations or translocations that are frequent, especially in AML. Thinking as a mesothelome as part of all the different epigenetic changes or histone modifications, gene expression changes of non-coding and coding RNAs and aberrant splicing, and I'm just ending here. But I think it's a broad topic, and I felt like every other week we learn more that we should be looking at so many different things and how they all go hand in hand with each other, similar as the cosmos does. And AML is, just as a little background, is a cancer of the hematopoietic cells. It results from the clonal expansion of undifferentiated blasts in the bone marrow. The outcome of AML patients, even though they improved over the past decades, are still very poor, with only roughly 30 to 35% of younger patients we classify as younger than 60, and only 10% of older patients achieving long-term survival. And AML, based on the WHO, is classified based on the morphology of the leukemic blast and associated dysplasia. Just looking through the microscope, what do we see? Surface and intracellular protein expressions and specific chromosomal abnormalities. But AML was one of the first diseases where, in addition to these features, in 2007, gene mutations were added to also belong to the classification of AML, and were found to be important for multiple different reasons. And when it was started to be brought up into the WHO in the early 2000s, only very few gene mutations were known at that time. Afterwards, larger genomic studies such as the Cancer Genome Atlas in 2013 were the ones who really revealed how diverse and how molecularly complex AML is. However, it is molecularly complex, but it's also non-random, and I think it's a very fascinating thing about it is that we see the same chromosomal translocations, the same gene mutations over and over again, and that we are following up on them. So, just in general, I think we can look at gene mutation as much as we want. And I think it's very important to think, why are we looking at them? What is the relevance? And I think there are very different relevances. Of course, there's a biologic relevance of a gene mutation, but there's also a clinical relevance, and both of them are overlapping. It's not that one mutation is one more than the other. But I I'd always like to remind myself of the reason why I'm looking at a mutation in a certain patient. So biologic relevance for me means that this is a mutation that is, for example, a genomic hallmark of AML, so perhaps able to contribute to disease initiation or modify the course of the disease. And I think this is like one of the very early papers from the WashU group that showed the idea which mutations might be like disease initiating and which ones come on later. The second one, and when I think of the clinical relevance, I'm trying to separate it into prognosis and into treatment. And for prognosis, these are mutations where we've based on older studies, the way older studies, because when we didn't have many other treatments, but what we call 7+3 intensive induction chemotherapy available, we would be able to say, if I'm giving a patient with leukemia this induction therapy, what is the likelihood if I have mutation A and mutation B that this patient goes into complete remission and also survives for a longer period of time? And these mutations that were validated in larger studies were then, for example, integrated in the most commonly used genomic risk classification, which is for us the European Leukemia Net Genomic Risk Classification. And I'm just showing it here for you as an example. It classifies from green, yellow to red patients based on their cytogenetic findings and their gene mutations into different risk groups that all have different outcomes. And I would have just up until a couple of years ago, I could have stopped here because every of our leukemia patients would have gotten the same treatment. But luckily, that changed over the past years. So by now, over the past years, we are also able to really start making treatment decisions because we have other treatments available than only intensive induction chemotherapy. So, for example, if we know that there are certain gene mutations such as TP53 that associate with a very poor prognosis, like TP53, you know, your chances of responding to the standard of care is only 5%, then you now have different therapeutic options that you could choose to do instead. And this is the real beauty about it, is that for leukemia, we now have the availability of different targeted inhibitors that were just approved. And because it's so important, this is just a snapshot of what we are doing on a daily basis for our leukemia patients. By now, you see here as an example, if I have a 45-year-old female patient, we just took it out of a patient I'm seeing here on service this very moment. We do the cytogenetics, but we are also testing for gene mutations, as you can see here, IDH1, IDH2. And why is this important? And this is how gene mutations have, over the past couple of years, changed our opportunities to do treatments. So I think my cursor is not working, but you can see in the middle that the molecular testing is one of the important factors that really guides treatment decisions, where I can add very specific targeted therapies based on the presence or absence of gene mutations. So this is super exciting. But there the question is, if I now have the opportunity, if I have now these drugs available, how can I make sure that people are actually getting this testing? Because for us, being at a major cancer center is obviously easy. But if you're somewhere further away, that may be like a big challenge. So I'm actually, I am personally not researching in that area how to do that, but I was very excited to see Dr. Mercuria's talk and see how many people think in similar areas, because one of my colleagues here, James Blatchley, has been trying to make it easier for our colleagues here in the outpatient setting, in more rural areas, to do this next generation sequencing testing for AML patients for these mutations. And he has been also using the NanoPer system. I'm not as much a technical person that I could compare the assets, but I think the idea of having this small and low cost instrument which is originally prone to have very high error rates, but similarly seen if you use bioinformatic tools to be able to model the instrument error and retain the neural network so that you're able to really reliably call these mutations. It's a very beautiful test that could be, used hopefully in the setting of clinical trials or just on an everyday basis to be able to offer patients not only the standard of care, but these targeted available therapies. And obviously, AML is just only one of the diseases. This can be hopefully, from lung cancer or other cancers, be a great additional way to get this information. So going from the small scale, I said we should see how we're moving away. This was an idea to look at single gene mutations, but going back to the cosmos of cancer genomics, and us knowing how mutations are influenced by gene expressions and by epigenetic changes and the other way around. There are, of course, also now very, very complex and more sophisticated approaches how to utilize this information in integration. The possibly most comprehensive approach for somatic tumor characterization that at least I know of is the Institute of Genomic Medicine Cancer Protocol. So what they're doing here at Nationwide Children's, and this is a protocol led by Elaine Mardis and her group and her team, is that they are taking tumor normal DNA from each patient along with RNA extracted from the tumor only. And the assays are, and this is routinely used for the care of relapsed and refractory children with cancers here at Nationwide Children's. So the assays are coupled with multiple analytical pipelines to track the cancer and to obtain a readout of the cancer biology that occurs from DNA level alterations, including the results of epigenetic alterations up until chromatin packing. And so they do basically a whole exome sequencing, the very high coverage where they're able to look at somatic and also germline mutations. They're able to do copy number variations and do a detailed clonality analysis. And then they pair it, as I said, with RNA sequencing of the tumor, where they are looking at gene expression, but also at fusion genes. And with all of this together, they are able to identify not only the mutations, but also the downstream effects that a mutation would be having in the way if it's deregulating a certain pathway or if it's like sending a certain signal. And so far, Nationwide has analyzed 115 patients. These were all relapsed refractory children with cancers, and they were able to successfully detect at least one meaningful medical change in 95% of these patients, which I think is absolutely remarkable when you think about it, when we're using regular panels, how often it is. And this is possible because they went ahead and didn't only look at fusions, but also really expressions and other variations. We have been trying this out also for AML patients, and this is just one example how we were trying to figure out what happened for a patient with AML between diagnosis and relapse, and because he didn't respond to any relapse treatment, and we were able to identify how the clonal evolution went and identified fusion genes that would be impacting, and at the end we're able to track back a certain pathway, and this patient is now going to be enrolled on a trial where the trial drug is going into this pathway treatment. And we thought, can we even go further, going away from a certain patient, but what is if we take an entire group? Because it's such a beautiful idea to use all these different tools together and kind of come up with a matched signal. So we chose to look into a molecularly fine subgroup of leukemia where all patients received the identical treatment. And these were patients with a good risk inversion 16 leukemia. And we're able, by the gene enrichment analysis, what you see down in G, the first hint we had that there was an increased histone acetylation pathway activation that was putting already one hint into an importance of this, but when we were looking at gene mutations that would be occurring and at copy number variations, we also saw that there were specifically histone genes affected, and all of those pinpointing to an importance of HDAC inhibition possibly for patients with a specific subtype of AML that are relapsing with their disease. And that's something that we're trying to follow up now with prospective studies. So I think this is, of course, a very broad tool that can't be used, but I think this combination of the different parts of cancer genomics is very fascinating. Now, this is a hard cut between AML and me philosophizing now next what we can do to make this cancer genomics from a single gene mutation testing up until the idea of doing a broader and really integrated genomic profiling to a broader group of people. And this, what you can see here, is actually it's not my thought, but of people here from Ohio State, namely Heather Hempel and also my mentor, Albert de la Chapelle, who thought it's now 15 years ago, what could they do to test every patient in Ohio, everybody diagnosed with colorectal cancer, whether this patient would possibly have a hereditary mutation, a mismatch repair gene mutation, that caused them to have or predisposed them to the development of a malignancy. And because why is this important? Because they already had that cancer. Their thought was, can we test the patient, identify if they have a hereditary predisposition, and then also do a cascade family testing. And that's exactly what they did. They had 50 participating hospitals, enrolled patients in all 88 counties. And based on that, between 2013 and 2016, OCCPI enrolled 3,400 adults, which was roughly half of the estimated early onset cases based on tumor data and registry data. And out of those patients, except 3%, all of them received the tumor and germline testing that they were supposed to get. And as a result, OCCPI was able to diagnose 153 Ohioans and 239 of their relatives with Lynch syndrome. For me, I thought the setup was great, that they were able to use local providers, but provide centrally navigated, I think that's the best way to put it, navigation of their care and order the testing that's supposed to be done and help the local providers out that are probably perhaps too busy or perhaps wouldn't know about it and reach out to the patient. Since that was so successful, we are now trying to adapt this for a second interest of mine that I'm very passionate about, which are young adolescent and young adult cancer patients. And we are trying to set this out for those with the most common AYA cancers, breast, colorectal, and brain sarcoma or heme malignancies. We're trying to use the exact same hospital network. And where the idea is that we're going to look first for a year how providers are usually ordering genomic testing for those patients and see how patients are doing, and then go into an advanced genomic profiling phase. And this should be done with virtual AYA navigator support. So where somebody is sitting here in Columbus, Ohio, and is reaching out to all these different hospitals and identifies the patients via access to their medical records that should be getting genomic testing, so that they would be getting advanced tumor testing with a panel over 650 genes, where we are partnering with a company with Tempus, and to do the full profiling. And then we will be able to do the result communication to them also virtually. We will also be doing, the idea will be to do also germline testing. Since they're young patients, they might be having a predisposition. And over the entire course of the study, looking how the patients are doing and if it changes the patient reported outcomes, and observe them after. And then my hope is that this would be something, if this is successful, and that this could be expanded in different ways over above Ohio or into much other ways. And I think if one thing came out of COVID that is positive, it is that we, I think, all learned how powerful it is that we can do a lot of things virtually, even though we are not in the region. And I hope with people getting more used to do virtual counseling, to do more virtual visits, this is something that we can really take advantage of in the future. This is just a somatic tumor profiling workflow, but since I'm getting over time, I just have a very quick summary which says that genomic testing in both small and large scale has the potential to really change the treatment and outcome of cancer patients. But we need to find a balance to enable appropriate testing for everyone, whether we need a gene mutation or an entire full integrative profiling, and that should be tailored to specific subgroups. And both centrally navigated and locally executed testing with virtual support, or fully local small-scale testing methods such as the nanopore represent options to broaden the availability of cancer genomic testing. Thank you so much.
All right. Thank you, AK, for a very interesting talk. And I want to thank all of our speakers, Dr. Tato, Dr. Rochford, Mercuria, and Dr. Eisfeld for a great session. And we have about 15 minutes to take questions from everyone present. And you can just enter them into the chat or to the question. And at this point, I'll start with I have a question. So the nanopore material that Dr. Mercuria presented, and the really fascinating work that AK Dr. Eisfeld has presented left me with the question of how we could apply such technology to resource-limited areas. AK, you discussed the relevance of clonal evolution and how our tools can pick this up and follow it, how the acquisition of somatic mutations can be detected and then acted upon with newly targeted agents. Knowing that colorectal cancer and acute leukemia, chronic leukemia are amongst the most frequent diseases or cancers in sub-Saharan Africa, I wonder if you or perhaps Zelalem could comment on how we could apply this next generation technology to benefit patients in those regions.
I can at least give it a start. I think, especially thinking of regions that are further away, such as sub-Saharan Africa, what is beautiful about, and I think both examples that you brought up are great examples. Like, since colorectal cancer, we don't have the need to look for 100 different mutations, which I think is challenging. But in AML, it's the same, because what we need to find out are targetable mutations, and we have a handful of them. And having such a device, such as the Oxford, like the nanopore or something similar, is great. But I do think it needs to be coupled, if one has it available, really with virtual support in some other way. I think in an ideal way to have something similar where one has providers that are going to the network and having like virtual support, be it from like not even medical people, but just people who are trained enough to say these are patients who should be getting it, and then having follow-up sessions where one then explains the meaning and like provides those parts. I think I think it's a beautiful way, and I do think there shouldn't be many reasons that would hold us up from not starting to do that.
Go ahead. Yeah. All right. Let me see it from a perspective of resource as well as from the technology itself. So if you consider resource-constrained countries such as in Africa, I think so. What a nanopore type of platform does is really give a chance for smaller labs or chances for areas which have really significant capital constraint in terms of buying these very expensive instruments to do some genomics work. In that line, from the experience I have with working with the company, they're very active in giving support and at the same time developing new diagnostic utilities. For example, the new LAMP core, they combined the LAMP assay, which we usually use for detection of now SARS-CoV from saliva and stuff, and combined it with the sequencing. So that workflow is get the sequencing cost really cut down to $10 to $20 per sample. So that's really significant, I would say. The other thing is the convenience, like really in terms of teaching, capacity building effort, really teaching Illumina sequencing platform in terms of laboratory library preparation is very extensive. So you can actually get engaged people into next generation sequencing very easily using the nanopore system. So even giving it a thought, what I had in 2017 when I started using nanopore sequencing, and now the advancement in terms of advancing the reads into having higher qualities in terms of throughput, getting a higher yield is significantly improved. So it seems these things contribute. Then in next generation sequencing, you have more data, means the cost goes down immediately. So it will give a chance to these third world countries to do next generation sequencing. So important. And specific assays for nanopore in terms of pathogen discovery. I'm not sure any assays for cancer and things like that, but for microbiology, there are several coming out in pipeline. So it will be a significant. So the good example is this is a technology that comes in 2014, 15 into the market. Now 20% of the SARS-CoV data from Africa is done using nanopore sequencing.
And the amount of DNA that's required to get adequate number of reads, could you comment on that?
Yes. So the company required 1 microgram of DNA, high quality DNA. That's one drawback of a nanopore system because it doesn't use any kinds of amplification or PCR steps. It's a native sequencing, which has an advantage by itself. It avoids these artifacts that could get into because of the amplification steps. And it's native DNA molecule sequencing, so you get what you give it. However, the input amount is really high compared to what is required for sequencing like Illumina. From my experience, we have titrated actually some samples and try to get a good coverage for bacteria as well as viruses. And for bacterial isolates, the requirement is now they have put it to 100 to 200 nanograms, but up to 50 nanograms of DNA. It's a very weird way of assigning quantity, I know, but that could give a very good sequencing yield. But because it is a direct sequencing technology, the higher is the better.
Sure. So I'm curious, Rosemary and Christina, perhaps you could comment when studying pathogens and like EBV or other viruses. How likely is it that from saliva or plasma that we could collect or isolate a microgram of DNA for the use of the nanopore technology?
I'll speak to that from the EBV perspective. That would be tough from the peripheral blood. From the saliva, absolutely. However, I think it'd be worth looking in Burkitt lymphoma patients because we know there's a high viral load in those patients. From tumor lysis or things like that. So we might have sufficient. I think it'd be worth trying that to see what we could get in the plasma because we know the viral load is quite high there. But from a healthy person, I don't think it would work for the peripheral blood. Saliva, yes, but not from blood. That's a real challenge.
Mm-hmm.
Yeah, I agree. It's definitely a big challenge. Most of the amounts that we deal with are much smaller than that. And really it's about timing, right? Especially for infectious disease. If you don't sample in the right place and you're not sampling at the right time, you're going to have more or less chance of actually being able to detect something. So that's a large part of it too.
You know, one of the nice aspects of nanopore technology is that it allows you to look at covalent modification of DNA, like, you know, methylation content, and some of the work that we're doing here and with the collaboration of Rosemary is actually assessing, you know, what's the nature of the DNA that we're detecting, and is it likely coming from a viral particle versus a free DNA in the blood or in the saliva. And that could have implications on the success or the efficiency of actually getting long reads, knowing which patients to go after, like Rosemary suggested. Perhaps, you know, the Burkitt's patients are the ones to focus on initially because of the high viral load in the blood and the saliva. But lots of really interesting opportunities to explore, and I think when we understand that, we can really make some progress. We only have about 5 minutes left, and I would like to just remind anyone in the group, if you have a question, to post it in the chat or the Q&A. I have an additional question for Dr. Rochford. You had mentioned earlier the ZPV3 variant is more commonly found, or at least that virus infects T cells more efficiently. Is there any idea why that is?
It's the EBV type 2 infects the T cells more efficiently.
Oh yes, I'm sorry, the type 2. Okay.
Yeah, but it's through a mutation in GP350. So it looks like there's a enhanced binding site for the CD21, and so we think it's that mutation of the GP350 that allows it to bind to the CD21 on T cells, which is very low level on T cells. It's there, but at a very low level.
Okay.
And so that's why. And I think, you know, that the point I was trying to make is that, and actually I like the galaxy picture, is that we can take a big broad look at all the sequences, but then we still have to dive in and try to understand it, bring it back to pathogenesis. So it really is an integration across from the first broad strokes that we take a look at, and then diving down in specifically to help us understand either etiology or disease progression or epidemiology.
Yeah, that's a great point. If I recall, your one slide had shown that the ZPV3 was also more commonly seen in type 2. Is that correct?
That's correct, yeah.
So it's interesting that there's a mutation affecting two key lytic, one early and late gene. And when I think about this, I wonder if this type of virus could be relevant biologically in terms of the peripheral T cell lymphomas and extranodal NK lymphomas.
That's what I'm wondering, whether you should start looking for that mutation in those lytic genes to see if they're there. I think that's going to be a really important question.
Okay, we're coming up on our time limit. If there's any last minute questions, jump in quick. Otherwise, I'm just going to thank our plenary speaker, Dr. Tato, for a wonderful plenary session. This whole session has been very interesting, and I think it's run the whole gamut from you know, pathogens to the human application, and just a real exciting session. I know Dr. Eisfeld jumped in at the last minute. She's on the acute leukemia service, so a special thank you for fitting us in today. Really appreciate that. If there are no other questions, we can probably move on, and I can hand this over to Kaylee.
Thanks, Dr. Baiocchi, and once again, thank you to our presenters. Very detailed and informative presentations. I am going to launch a poll for this session. To our attendees, please complete this if you're willing. This helps us evaluate how we're doing and how our information is presented. especially given our virtual format. I will leave this up for the next 10 minutes or so. But instead of making everyone wait, we do have a very special session to close today. A number of our speakers have mentioned the need to collaborate and the challenges in working across disciplines to really practice One Health. And there are a lot of opportunities there. So we do have a very special guest who will be introduced by our own Donald Omothuna. I will turn it over to Donald.
Hello, everybody. I've been enjoying the sessions here over the last couple of days. So my own area is in ethics, and particularly around research ethics involving humanitarian contexts. And through that, it's been a real privilege for me to encounter our speaker, Dr. Cesar Ature from the University of Ghana. He gave a fascinating presentation this summer at the World Congress of Bioethics, and I thought that this would be a really excellent addition to our conference. We've seen a number of different intersections throughout the couple of days about the importance of the humanities coming alongside with the science and the technology. And I think Cesar will be able to do this for us in a really great way. Cesar is in the Department of Philosophy and Classics at the University of Ghana, and he's also a fellow at All Souls College at the University of Oxford. He is a bioethicist and a philosopher, a thinker, but he's also a man of action, and he has helped to found an NGO called Amicus Onlus, which seeks to bring healthcare to rural populations in the coastal areas of Ghana. So it's a real privilege for me to introduce Cesar, and he's going to talk to us about solidarity and how that has an important role in the types of developments that we're talking about. Over to you, Cesar.
Great. Thank you very much, Donald. I'll just start off by sharing my screen. I hope this works. Let's see. Is it visible?
Yes, it is. If you could put it onto the presentation mode, then we'll see it better.
Okay, let's, yeah, it's excellent. So, well, thank you very much for giving me this opportunity to share some of the thoughts we've been reflecting around on the concept of solidarity, health, and One Health, really. I think COVID has given us an opportunity to rethink our vision around human health and the social determinants. And I'm really grateful for this opportunity, Donald, Amanda, and all the organizers. And I'll just share some ideas, and perhaps at the end, there will be time for a question and answer session, and we'll be able to take it from there. Trying to say what I'm about to say, and especially as a philosopher, in about 20 minutes is not going to be easy. And so I'll just give you an overview of what I intend to say, with the hope that I will be able to say it, and then we'll move on from there. So basically, I'm talking of 5 basic ideas that I will be presenting. And I'm trying to draw here from the African traditional and philosophical way of thinking, because I think as we're moving ahead, we need to broaden the global conversation around health. We're now seeing that we're so interconnected and so interdependent that one single framework alone cannot embrace all the challenges that we're facing. So I'll be drawing a lot from the African philosophical tradition. So first of all, I just want to look at the question of persons and personhood. I mean, at the end of the day, we're talking about human health, but what is a human being and how do we conceive of human beings? Then I'll introduce the philosophical concept of moral standing, which is what makes us owe duties towards certain beings. Then health and healthcare. How do we conceive of healthcare within this framework? And if healthcare is an individual right, how does solidarity actually fit into it? Then, are we really, and this is a question that I'll raise at the end, the relationship between our, as it were, prerogative to express autonomy and also live in solidarity. Are these two things antithetical? Can they be brought together? So I'll just move on quickly and talk about, our conception of persons and personhood. Now, there's one, I'd like to start off with an epistemological consideration. In general, the way we study science and the way we study even the humanities is that when we're trying to define something, we try to look for the constitutive elements to be able to define the object. So even if we're trying to define water, what we try to do is to look at the fact that water is made up of hydrogen and oxygen, and at the end of the day, we have, let's say, a scientific definition of water. However, in the African tradition of thinking, relationality is also important in defining an object. I'll just give an example. Recently, I had the opportunity of traveling near to one of the towns where, in Ghana, where people actually live on a lake and they live on stilts. Now, these are people whose relationship with water is quite different to the relationship many of us have with water. Learning to swim for them is equally as important as learning how to walk, if not more important. Now, in their perception, understanding of what water is, there is something different to how other people may perceive what water is. What I'm trying to say is that, yes, in trying to define an object, we need to take care of the constitutive elements, but at the same time, we need to also to consider the relationship we have with that object. I could give other examples. If you live in the desert, your relationship with water is different to somebody who lives in a green belt where there is a lot of water. Now, this is quite different because if you look at the Aristotelian categories, when Aristotle talks about, the ousia, the essence of something, he doesn't include the category of relation. Relation is seen as accidental. However, in this framework that I'm going to present, relation should be considered as part of the essence. And under this approach, what we see within this framework of thinking is that even in the linguistic approaches, for example, within the African context, when you talk about trying to say who is a human being, in the Bolsa conception, we talk about Nurbi, which means a daughter or a son of somebody. Therefore, our being, in its essence, is actually grounded on relations. I was just listening to the previous session, and we're seeing all the advances that are being made in terms of DNA, in terms of neuroscience and all that. And philosophically, this is actually pointing to how much we are related, because at the end of the day, even our constitutive elements that define who we are received. And this also goes on to the ontological grounding that we find, for example, among the Akan people who say that the human being is made up of three elements, which is Okra, Sumsum, and Honam, and this would translate into a soul, a spirit, and a body. However, all these elements are inherited from the parents, or they are inherited from the mother and the father, and the Okra, which is supposed to be the divine spark and the principle of life, is actually inherited from, and let's say, a cosmic or supernatural source. The same concept is repeated when we move down towards the southern part of Africa, and we're talking about, Ubuntu, which is, I'm sure many of you are familiar with the notions of Ubuntu, the fact of being human, human dignity, and it simply claims that we actually become persons through other persons. So the idea of actually trying to think of a human being as a disengaged reality, as an atom, is intrinsically contrary to the way African philosophy perceives of a human being. And if we take one step back, really, this is nothing new, because even Western philosophers like Charles Taylor talk about the fact that the disengaged self doesn't exist. I mean, if each and every one of us is born out of relations, and even before we start communicating, we already need a relationship, which is a common language. And without relations, we can't even communicate with each other. So it is important to bring that category of relationality into our conception of what we are, and therefore we are interconnected. Now, this also has normative dimensions. The normative dimensions means that once we are intrinsically related, we do need of some form of community or some context within which to live. A human being, as Aristotle would have put it, it's only a beast or a god that can actually live without any social relations. It is impossible for a human being to survive without relationality. Now, there are various ways of conceiving our relationality. There are some who tend to insist a lot on our communal dimension, perhaps to the disadvantage of the fact that we are individuals. And the dominant model that we tend to use, which I call the Legon tradition, is that model which we call moderate communality. A moderate communality means that the community, the society within which we live, is actually at the service of our individuality. And it is at the service of our individuality, and in expressing our individuality to its full, we also contribute to the growth of the community. So it is not necessarily an antithetical relationship between being communal and being an individual. Every individual, through their identity, contributes to the wealth of community, and it is the duty of community to make sure that every individual is able to flourish. There is a beautiful Akan saying which says that the community is like a forest. Seen from afar, it seems like a cluster of trees and it seems the same. But once you get close to it, you see that it is made up of different trees and each one has their own identity. Now, at the center of this, what we do have is that the important element is that it is a kind of humanism that we're trying to propose here, a humanism whereby human value, the value of being human, is what is at the center even of the interest of community and at the interest of each and every one of us. Just one point before I move on to the next slide is this. There is a difference in this conception with other communal conceptions that I would like to underline. What I'm trying to say here is that our being related, our communality, is actually a given. It is not fruit of a contract. We have other traditions that have tried to base our social existence on the idea of a social contract. But that idea of the social contract assumes that we actually exist without relation, and then we enter subsequently into the relations. What we're saying here is that our act of being, our act of coming into being is actually relational, is already relational. And therefore, communality is not a contractual utilitarian commodity or utilitarian arrangement, but it is actually a given, and a given that is part of our specific difference as human beings. Now, let's look at a second concept of, around being human. One of the characteristics when we talk about moral philosophy and we talk about normative thinking is that, yes, human beings have rights, but this is based on the fact that, human beings have a moral standing. And beings that have a moral standing are beings that are worthy of consideration in our moral decision making. So you just do not treat them the way you treat other objects. And beings that have a moral standing impose duties on us simply because of who they are or what they are. I could decide for any reason, right or wrong, to perhaps even scratch, let's say, my neighbor's car because he keeps parking in my parking lot. But in doing that, I'm not offending the car because the car hasn't got a moral standing. But my neighbor does have having a moral standing and not having a moral standing. Now, with regard to humans and with regard to the notion of moral standing, within this framework of thinking that I'm presenting, there are two characteristics that are important. On the one hand, we have what we call an object moral status or moral standing, which means that a being, simply because it is, deserves moral consideration. And this is something that in the African framework is actually attributed to all human beings, irrespective of their capacities, irrespective of their achievements. Every human being does have an object moral standing, and for that matter needs to be respected, and for that matter we owe duties towards that human being. There is, however, a second dimension to moral standing, which I call subject moral status. And this is also accorded according to the degree to which a being contributes towards the good of humanity, towards the communal good. Now, one would find, even in the literature among African philosophers, that sometimes people say personhood is to be achieved. And when they say personhood is to be achieved, they're looking at the subject moral status, which means that as a person contributes more towards the communal good, towards the good of everybody, the person acquires a greater moral standing. So a full moral standing would be to have a high level of subject moral status by being somebody who's kind, compassionate, contributes towards the welfare of everybody, and also by having an object moral status, which is just a question of being human. The interesting environmental implication of this particular vision is that there are objects, natural objects, that are accorded some form of moral standing even within this viewpoint, so that these objects, even though they may not enjoy the object moral status which human beings enjoy just by nature, but because of their contribution towards the humanistic vision, they are given a moral status. And that is why, for example, the third point on the slides, looking at one of the communities here in Ghana, the Bolsa tradition, the land is considered to have a moral status. And when evil is committed, it is believed that the earth has been offended. And therefore, reparation needs to be made to the earth. If you go to the coastal areas where people depend a lot on the ocean, there is almost a veneration of the ocean. So here you see a different form of environmental ethic at play. The environmental ethic is not just a question of utilitarianism, that these things are useful for us, and for that matter, we need to protect them for future generations. But because these are objects that actually allow us to live as human beings, they merit respect. They have a moral status, they have a moral standing, and they are capable of actually being offended. And that is why we need to treat them with respect. And when we do not do so, we need to do acts of reparation in order to make sure that this environment, these natural objects actually allow and contribute to human flourishing. Now, so far, I've introduced personhood, I've spoken about moral standing. Let me introduce a third concept before I start bringing all of them together. The third concept is about health. Now, health in the African tradition is viewed not just as a physiological well-being. And here I cite, Peter Omonjelele, who talks about health as a state of mental, physical, spiritual, and emotional stability. Therefore, health is seen in a holistic way. And what we actually noticed, and many of the previous speakers mentioned work in Africa, is that people in Africa, for example, would go to a hospital to look for cure for their diseases, a physiological cure. Yet at the same time, they will be looking for some form of spiritual or metaphysical cure for their illness as well. Why? Because the vision of health is not just a question of a body functioning well, but being in a condition to be able to live a flourishing life. And that flourishing life means actually living in harmony, and means living with emotional stability and having healthy relations. And that is why Badejisen, for example, a Nigerian philosopher, talks about the condition of ill health is the idea of disease. He breaks the word disease into two, is dis-ease. And therefore, it is the question of being in a state of not being at ease. And that condition of not being at ease is not only physiological, but it also has metaphysical, sociological, and psychological dimensions. If we accept this vision of health, then what emerges is that health is no longer just an individual good, because if health has social dimensions, if it has psychological dimensions, if it has relational dimensions, then our health is interconnected. And I mean, if our health is interconnected, then health is also a communal good. And this leads us to the next point that I'm going to be developing, which is the fact that healthcare as a framework is necessarily solidaristic. It is not just a question of an individual being healthy, because I mean, let us try to imagine today, in the context of COVID-19, if everybody just decided not to care about others, how would the world be? We're being asked, healthy people are being asked to practice some social distancing, to wear masks, because we owe a duty to the well-being of everybody around us. And that means that the duty bearers actually of my right to health and your right to health is not only the government. The duty bearers are also each and every member of the society. We are all duty bearers of other people's right to health, just as they are also duty bearers of our right to health, because our health is interconnected. And there is a beautiful saying among one of the ethnic groups here, which says that, I mean, if you're looking for a therapy for your health condition, just go and sit by the road that leads to the market, because the market is a place where many people go to, and you will be sure that people will stop by and offer you help or advice, because everybody feels they are a duty bearer to your being well. Now, this type of vision of health means that we cannot talk of healthcare without looking at the social determinants of health, looking at inequalities that can actually put some people or some members of the community in a condition of disfavor when it comes to health and healthcare. So, if health is a shared good, both individual and also communal, then a solidaristic framework is perhaps the best way to approach healthcare. And I mean, perhaps if you look at some of the European traditions, countries that have national healthcare systems, one can identify at the core of these healthcare systems a solidaristic model, because at the end of the day, everybody is taxed, everybody contributes, but it's not everybody who takes the same. But it is based on the assumption that the healthcare systems are, I mean, our health is interconnected. Now, I'm just going to look at the concept of solidarity, some characteristics of the concept of solidarity, and we see how this fits into healthcare. I'm just borrowing a definition here from Prinsak and Bui from 2016, where they talk about an enacted commitment to carry the costs, and talk about financial, emotional, and other contributions of assisting others with whom a person or a group of persons recognize similarity in a relevant aspect. Some of the key concepts of this definition, I'll just run over them quickly. One is this idea of an enacted commitment. You see, when we're talking about solidarity, sometimes there's a vision that sees solidarity more as aspirational. But solidarity also has a normative dimension. It is enacted. It is not just mere empathy. And there's a beautiful paper, Journal of Political Philosophy by Ashley Taylor, who makes the distinction between a normative or robust philosophy, a solidarity, and an aspirational solidarity. And the type of solidarity that I'm referring to here is a normative view of solidarity. Now, the idea of sharing, the definition talks about actually carrying the cost. I prefer to use the word sharing the cost because at the end of the day, the origin of the concept of solidarity, which comes from our modern concept, which is derived from the Roman law of the obligatio in solidum, talks about solidarity more as sharing responsibilities, such that if a group is considered a solidaristic group and somebody in that group perhaps owes, even after the demise of that person, the other members of the group will also be held responsible. That's the obligatio in solidum, which means that a solidaristic group has a sense of we-ness. And the sense of we-ness means that success or failures of the group or of a member of the group are also successes or failures of the whole group. We see this too in some other solidaristic groups. Take, for example, even trade unions. When some members start to lose their jobs, it would look as if the group is actually failing because some members are losing their job, because the success and failure of members is linked to the success and failure of the whole group. Now, solidarity is also in assisting others, but there is a distinction here between solidarity perceived as solidarity towards and a solidarity with. Solidarity towards means sometimes it's perceived as helping other people. But the normative and robust form of solidarity that I'm proposing here is that solidarity which means to stand with. So if, let's say, during the Black Lives Matter protests or demonstrations or in marches in the US, a white person decides to join the march. Now, there are two ways of joining that march. One way is to say, I'm siding with Black people, which is solidarity towards Black people. Another way is to say, I'm with the Black people because they are part of our community, and whatever evil is done to any person within our community is also done to me. And this is not what our community actually stands for. And this is solidarity with. And this is more important. This is a stronger form of solidarity because we stand together. Now, this then leads to the other point, which says that solidarity, unlike charity, is actually based on recognizing similarity with the other persons in a relevant aspect because we are in it together. It's interesting that one of the phrases that has been repeated a lot during the COVID pandemic is we are in it together, because we suddenly realize that we actually have a lot more in common. Now, this type of solidarity is based on similarity, unlike some other models of charity that are based on almost like pity or compassion towards somebody who is less fortunate and is different from us. So I can, at Christmas, I can send money to starving children in Africa, but they are different. Their condition is not my condition. That is more of a charity kind of approach, a type of charity approach. How am I doing for time? I'm not seeing the messages, so I can't read the...
Yeah, we got a couple of minutes left, Cesar, if
Okay.
we could leave time for some questions too.
Good. So I'll just mention the idea of solidarity and autonomy. Autonomy actually entered the philosophical tradition through Kant, but the autonomy that Immanuel Kant was referring to was to give laws to oneself, autos nomos in Greek, right? So we're giving norms to ourselves. But he was saying that as a reaction to the Christian ethics which were imposed, or as it were, which were drawing from a heteronomous source, drawing from revelation. But even Kant, in his autonomy, or his model of autonomy and autonomous morality, talks about universalizing. We need to consider others, because what we're doing, if it is to be right, is something that we should be able to universalize. So autonomy, taken seriously, is about deliberation and making right choices, because autonomy is not just simply having the possibility to choose, because we can actually make wrong choices. And I think we're all old enough to know that we have made wrong choices before, and we could still do the same again. The idea here that, just to sort of like synthesize, with the diagram that you see there is an Akan symbol from the Ashanti people in Ghana. The name is a bit long, but here we're just talking of conjoined crocodiles. These are crocodiles that have one stomach and two different heads. But what this model is trying to say is that even though our heads may be different, even though we may have different opinions, our common humanity, which is represented by the common stomach, is what brings us together. And the effort of morality, to a large extent, is an attempt to harmonize these conflicting interests through adjustment and adaptation, given that at the end of the day, what we are all interested in is in promoting our common humanity. This is the goal. And once we accept that, because it is in our essence to be related, because we are related beings, then that tension that is often exaggerated between solidarity and autonomy can to some extent be diffused, because I can actually choose autonomously to be a person who lives in solidarity, and that is a better choice. So practicing solidarity is not necessarily antithetical to living in autonomy. And I'll just stop there. Thank you.
Thank you very much, Cesar. I think it's very interesting to see the way you developed the distinctivenesses that we see in some of the African philosophy. So we have a few minutes for questions, and people are welcome to put those into chat, and I'll relay them. Or if you would like to raise your hand and speak out the question. And maybe to get us started, I know you've done a lot in research ethics, and particularly within Ghana and the Global Forum for Research Ethics. And I wonder, have you considered how the African perspective comes to bear within research ethics? Because I think that tends to have a very individualistic approach from a Western perspective, especially around consent to treatment or consent to participation and ensuring the autonomy of participants. Is that something that you've applied your ideas to, like you've discussed here?
Yeah, you're perfectly right. This is a very good question. It is something that we are currently, struggling with, because on the one hand, we work with international collaborations, which have a model of informed consent, autonomy, and at the same time, we also have the indigenous traditional models. Let's take, for example, just this week we had a workshop where we're talking about informed consent in clinical settings, where you actually, especially maternal health care, where at the end of the day, when a patient or a person is informed about their rights and the fact that they have to give consent, the person will tell you, no, I will need to consult, because my health, and especially my reproductive health, is something that we need to discuss within a family context. Now, these are things that are also, these are things that come up quite a lot. And even with regard to research ethics, the insistence on autonomy and sometimes runs into difficulties. And we notice, I've even given this example in other settings, we go out to do research, and if we're following the international protocols, we're looking for people to fill the informed consent forms and sign. And at the end of the day, and I remember this from a mental health research project we did a year ago, the participants were not so concerned about all the clauses of our form, but what they really wanted to know was, why are you doing this? Why are you researching into, they call mad people here? Why are you studying them? And when we were able to explain to them that at least our end is to be able to find ways to assist people, they accepted to be participants in the research. This is something that has several implications. It also means that the way we consider research participants needs to be different. We need to consider them not just as objects or people who just give a consent, but as people who are actually interested and investing in the finalities of the research. This therefore means that in sharing the benefits and in communicating the results, we owe a moral duty to let them know. But this is, we could talk about, we could get into a whole talk about research ethics. Yeah.
Okay, thank you. Does anybody want to ask a question? Okay, well, here's...
Yeah, this is Kaylee. I have a question just in terms, there's one that came through. I'll go ahead and let this comment go through first, Donald.
Okay, I'll read it then. So this is from Nicole. I loved your presentation, Caesar. I agree with a lot of what you said. I'm a student in the community health worker training program. People do thrive when they're part of a healthy community. When it is like a family supporting and caring for one another, and recognizing that when one suffers, it affects the whole community. We do have a moral duty to one another. So I guess that was a comment and a thank you to you, Caesar. So Kaylee, do you want to go ahead with your question?
Yeah, it built off the idea of the comment. In the U.S., we are so individualistic. We have a completely different mindset. So as a science community, as people collaborating across countries, what can we do, or how do we motivate people here to take steps to be more community-minded in our work?
Well, I wish I had a straightforward answer. But I think, and this is a future project that some of us are working on, many Western countries are increasingly becoming polarized. And it's not good for the countries. They're becoming more and more even difficult to govern. And a concerted effort in terms of education from the early levels to insist on the fact that we actually need each other, I think is part of an educational program that is needed. Our societies need to express more cohesion, because at the end of the day, the things that actually divide us are fewer than the things that unite us. But these things need to be reworked into the educational system. Secondly, the models that have been dominant, at least in the 20th century, and have been exported around the whole world, have generally, intellectually, especially in science, have been generated within the European or the American context. And I think it is beautiful that, for example, you guys have invited an African to talk to an audience in Ohio. We need to broaden this conversation to begin to realize that there are other ways of looking there are other ways of actually looking at things, and there is not just one way which is right. There's one beautiful saying by a theologian, Swiss origin, Hans Urs von Balthasar, who says that the truth is symphonic. It is through the various melodies, the various ways of thinking, that we can actually arrive at something that is harmonious for all of us.
Well, thank you. I think that's maybe a very apt place to leave our discussion. I would certainly love to keep talking with you, though. We've had a few other comments just thanking you for a really great talk and noting that it was very timely and relevant in the context of One Health. And certainly I think that comes across clearly. I really liked your point about concern for the environment, not just being a utilitarian kind of way of continuing to get stuff for us, but a way of actually truly respecting the environment and non-human elements there. So thank you very much, Caesar, for joining us. It was great to have you with us. And so I'll turn it over to Kaylee to let us know about any other concluding comments today.
Thank you very much and all the best.
Thank you. We have one ask our panelists if everyone would be willing to turn their videos on for a moment so we can get a screenshot to show what our virtual conference looks like. I'm sorry our attendees don't have video controls, but we would like to have that opportunity to capture everybody online. But other than that, thank you everyone for sticking with us, for participating, for your questions. We will be back here tomorrow for the final day of our conference. We'll be connecting with the same link, and we look forward to seeing everyone there. Thank you again, everyone.
Thanks, Kaylee.